Welcome to Jamie's Crash Course Immunology. I'm going to attempt to teach you everything I think you need to know for the CRE while keeping it high level enough that you don't need to get bogged down by the details. I will expand on certain things in red that I think can be helpful to your understanding, but I'm going to give you a high level summary at the end to kind of just give you something that you can walk away with and put on your CRE and hopefully get marks. Don't quote me on that. We'll see what happens, but this is what I think you need to know in simple terms. So I have told you about the innate immune system and the adaptive immune system. So as a recap, the innate immune response is your first line of defense against pathogens that infect your body. These are the cells that are going around and they are just monitoring your body for different things. And they can mount immune response very quickly, like within hours, basically. Now, when your innate immune defenses are overwhelmed or way down the line, like 5 to 7 days, I'm talking after infection, you can get your adaptive immune system to kick in. This is your second line of defense when your innate immune cells are overwhelmed. This response is also more specific to the pathogen that is affecting you because it needs time to understand the pathogen and to create specifically antibodies that are effective against very specific pathogens. The adaptive immune system is also responsible for your immune memory. So you can get memory T cells, you can get memory B cells, which store the memory of the pathogen that they encountered and can be reactivated in the future. And this is why you see people getting sick less frequently as they get older, because they have a memory to these immune cells. And by the time that the infection has started, your immune system is already ready for it. When your adaptive immune system kicks in, that's when you start to really feel the symptoms of things. You're starting to feel real crappy. Okay? Anyway. So, let's start really basic. Macrophages are one type of immune cell. They're an adaptive immune cell, and they just go around eating things. And I guess the way that you can remember it is like, they're like Pac-Man. They just go around and eat pathogens. Mac-Pac. Super easy. We also have mast cells and basophils. So these are granulocytes. So they contain these little granules. You can think of them like the little microbeads that you get in like soaps. When these release, they are inflammatory. So histamine, for example, or leukotrienes. These are inflammatory molecules that are what's contributing to your actual anaphylaxis symptoms. So what happens is these cells have high affinity for IgE antibodies that are produced by B cells, which we'll get to in a minute. But these cells are coated in these things, and when they encounter the specific pathogen that they don't like, so let's say it's a peanut, it sends a chemical trigger that is going to trigger these mast cells and basophils to basically split open and release these histamines and these granulo whatever, like to degranulate basically. So those are two of the cells that are specifically involved in like allergies and asthma, I believe. These are like in your type 1 hypersensitivities. I also mentioned eosinophils. These are another type of granulocytes. They're not as important, I don't think, because we don't really mention them, but they are involved in asthma. More specifically, eosinophils are involved in worms, so parasitic infections, wheezes, like asthma, allergies specifically, and weird diseases, which is a whole host of other things that I'm not going to get into. I don't think it's necessary that you really know this. So these are some of your innate immune cells. This is not all encompassing. We have other things like neutrophils, basophils, NK cells, what have you. But these are the ones that are important for now. We also have something called dendritic cells, which are also an innate immune cell. But these are your linker between your innate immune response and your adaptive immune response, because these are antigen-presenting cells, and there's only specific types of cells that can be antigen-presenting cells. That's macrophages, dendritic cells, and B cells. So basically this, and this all have the role of presenting antigens, which could be like a little piece of a virus, a piece of a bacteria, a piece of a pathogen, or an allergen, like a piece of a peanut that we don't like. They present them to T cells. And they do so via something called MHC class 2, which you can think of as like the platter for an antigen. I don't know that it's fully necessary that you understand and name MHC class 2, but if you understand it, like it obviously is a little bit better for your understanding of immunology as a whole. So yeah, they will basically take these antigens and get them into the cell, and they will like chop it up into fine little bits that can then be presented in a way that allows them to be presented on MHC class 2, which is like your platter. So I guess you can think of dendritic cells like the chef, and you can think of T cells like Gordon Ramsay. Gordon Ramsay says to your dendritic cells, to the chef, go tell me what's out in the kitchen, make me a meal with any of the ingredients you find. Your dendritic cell goes out, goes and looks through the kitchen, through the fridge, through the whatever, and starts taking these antigens or things that it finds, chopping them up, turning them into a nice little meal, and then presenting it on its little MHC class 2 platter, and giving this meal to Gordon Ramsay. And Gordon Ramsay can either love the meal and say, fantastic job, or he can spit in your face and call you a donkey and say, this is contaminated with a pathogen or an allergen that we don't like because Gordon Ramsay's allergic to peanuts, for example. So T cells have a few different functions. Gordon Ramsay has a few different functions, I guess. Can be either CD4 or CD8. So CD4 are your helper T cells. These are the cells that are going to go and stimulate other cells to do their job better, to get rid of pathogens better. Or they can turn into CD8 bloodthirsty killers and just kill virus-infected cells themselves. Now CD4 and CD8, why do we call them that? Well, these are cell surface markers. Cell surface markers are unique to every cell. They help to define what a cell looks like. You can think of them like the eye color, the hair color, the things that make the cell, and they define its purpose and they define its function. So all T helper cells are CD4, they express this CD4 marker, and all CD8 cells express the CD8 marker on the cell surface. And so I want to kind of break off here. I think it is important that you know these two types of helper cells, CD4 helper cells specifically. We have Th1 and we have Th2. Th1 is cell-mediated immunity. So these T helper cells help macrophages. They will stimulate macrophages to basically go and eat pathogens. This is your type 4 hypersensitivity response. On top of that, these Th1 cells will release something called IFN gamma, which is like the cell signaling molecule. It's pro-inflammatory. It's only important that you know this because it's important to know this for TB when you have this like IFN gamma assay that you can use to determine like a TB infection, because that is a type 4 hypersensitivity, if I'm not mistaken. Yeah, it is. This is cell mediated because we're dealing with an actual cell. That's why we call it that, like a macrophage cell. You also have type 2, Th2 helper cells. This is humoral immunity. Humoral meaning referring to B cells and their production of antibodies. That's what humoral means. And so these guys, Th2 cells, are the ones that are involved in allergies in your type 1 hypersensitivity responses. So they're the ones that are going to stimulate your B cells to produce IgE antibodies. B cells produce all kinds of antibodies. One of them is IgE. These are the allergy-specific ones. And so the cool part about antibodies is that literally every single antibody is specific for a specific pathogen. You can create antibodies to literally any kind of pathogen. You can create antibodies to a pathogen that comes from freaking space. That's how specific they get. There's billions of different combinations of antibodies. But they all fall under the major classes of like IgE, IgA, IgG, whatever. All you need to know is that IgE is specific for allergies. And so these IgE antibodies, they will attach to mast cells and basophils, which have a high affinity for them. And so they coat the surface of these cells. And so then the next time you encounter this antigen or whatever, this one, like an allergen, like a peanut, and it's the correct antigen for the type of antibody that's here. So if you have a peanut allergy, like let's say it's a peanut antigen, that's what's going to stimulate your mast cells to degranulate. So I'm going to give you a high-level summary right now. What you need to know. You need to know these guys. These are involved in allergy, okay? IgE specifically, mast cells, basophils. What's going to happen is your dendritic cell you can say is going to present an antigen, an allergen specifically, to a T cell. You don't have to necessarily say that it's MHC2 if you don't remember that. It's going to present to a T cell. The T cell is going to stimulate B cells to produce IgE antibodies. These IgE antibodies will bind to the surface of mast cells and basophils and coat them. The next time that you see the same allergen, a peanut or whatever, it's going to bind to this antibody and then stimulate the mast cells and basophils to release their contents and degranulate. And that degranulation releases histamine or leukotrienes or whatever, and those cause inflammation. Okay? That's your type 1 hypersensitivity. Your type 4 hypersensitivity, the one that's involved in tuberculosis, is you get your T cells. These guys are going to release IFN gamma, which is pro-inflammatory, and also stimulate macrophages to basically increase their activity and create that granuloma and everything. Okay? And that's done through your T helper cells. I guess you can be specific if you want. You can just say T cells, or you can say Th1, that's the one that's involved in type 4. Th2, that's the one that's involved in this allergy pathway with B cells and these guys. You can just say T cells, okay? I hope that makes sense. In general, if you want a little more detail, you can say how cells talk to each other. And the way that cells talk to each other is through cytokines. Anytime a cell is stimulated, it's going to produce signaling molecules called cytokines, that will either increase inflammation or decrease inflammation by stimulating other immune cells. So, for example, a T cell can either secrete pro-inflammatory cytokines that stimulate B cells to produce antibodies, or they can stimulate anti-inflammatory cytokines that stimulate B cells to don't do anything, basically. Whether or not it's producing pro or anti-inflammatory cytokines, you can think of cytokines like the language that cells are using to communicate to each other. It depends on the type of input that the T cell itself is receiving. So if we're in this pro-inflammatory state where we don't like a peanut, we don't like this, we don't want to do this, we're going to release pro-inflammatory cytokines. When we've cleared our infections or allergens or whatever, we're going to release anti-inflammatory cytokines because inflammation is inherently bad. Inflammation is not there to help your body out, to keep it, to make everything good. It's there to kill the pathogen, and in doing so, it can harm the body. And that's why we get all kinds of problems like ILD and things because of inflammation. So, that is that. Other things that you can know. Interleukins are a type of cytokine that you might see thrown around. You can see like IL-1, IL-8, IL-6, IL-21, all kinds of things. These are just a specific type of cytokine that's produced by leukocytes, which are white blood cells, which are like literally any of these. But that is not necessarily to know. The other thing is IFN. So you have IFN gamma. That is another type of cytokine, and these ones are specifically acting as alarms in viral infections. Although TB is not a viral infection, you typically see interferons in viral infections. That's just for your knowledge. I would like to explain MHC class 2 and 1 because I think this will actually be helpful for you, but you don't need to know this. All you need to know is that dendritic cells present to T cells. So MHC2 is what dendritic cells use to present to T cells. And dendritic cells themselves are a type of antigen-presenting cell. So these are the cells that can take an antigen and show it to a T cell. And other antigen-presenting cells are macrophages, dendritic cells, as I mentioned, and B cells. So any one of these three presents to the T cell using MHC2. We also have something called MHC class 1. So this is another type of platter, basically. But these platters are specific to... Well, they're not specific at all. They're actually found on any kind of cell. They're found on all cells in the body. Every single cell in your body has MHC class 1. You can think of this as like the defining feature that makes the cell a body cell. So if a T cell goes around and it encounters a cell that does not have MHC class 1, it is going to recognize it as a foreign entity, basically, and attack it. So I think you would see this in the case of like transplant, because they have MHC class 1, you know, like a transplanted lung is going to have MHC class 1, but it doesn't have your MHC class 1. So sometimes you get rejection of the transplant. So how do these work? What are they actually showing? MHC class 2 shows things that are going on in the external environment. So they are going to show like antigens or pathogens that are found in like the blood or the extracellular spaces between cells. That's what they grab and they put on their little things. That's what cells are grabbing on their little platter, the MHC platter. MHC class 1 is going to tell you what's going on in the internal environment of a cell. So a cell is for example, in the case of a viral infection, a virus infects a cell, and the cell can take a piece of that virus and basically chop it up and make it presentable to put on its MHC class 1, which then when a dendritic cell comes by and basically like feels around with its little things for the MHC class 1, it will feel this little piece of virus that's inside a cell and say, uh-oh, we got a problem here because there is a virus inside the cell. And then it'll go to a T cell and show it to the T cell on its MHC class 2, because it's an antigen presenting cell, and say, I found this in the cell. And then that's how you go and kill off the cell. And this is what your CD8 is going to do. It's going to go kill that virus-infected cell. MHC class 1 and 2, these are named major histocompatibility complexes. We found these in mice first. In humans, they can also be known as human leukocyte antigens, but we call them MHC 1 and 2. We don't really ever see people call it HLA. Don't worry about any of that. One other thing I want to talk about is Ig antibodies, basically. Ig meaning immunoglobulin. AEGDM is just referring to the different chemical arrangements that these antibodies can take to basically make up their structure. So there's 5 different types, obviously, as you can see here. IgM is like your default. These are like your short-term protection antibodies. They're going to like neutralize, well, antibodies are neutralizing in general, but they're going to neutralize the threat first. They're not very good for long-term protection because they're not super specific. IgG is the most common antibody. This is what IgM becomes. And IgG are more specific for the type of infection and pathogen that you're experiencing. So basically you get like B cells that when they're developing in the bone marrow, which is like the center of the bone, that's where all your cells are created, it starts producing IgM, but then it starts to develop instructions that allow it to turn these IgM into IgG, and so it starts producing IgG, okay? IgE is the one that is produced in allergy. IgA is often found in your mucosa, so think of like the digestive tract or like the respiratory tract, where there's just a lot of mucus. And then IgD, well, we have a little bit of it, but no one knows what it freaking does. So don't even worry about that. I think that about covers it. I hope this is helpful for you. Don't forget the things that I told you, okay? Dendritic cell presenting to T cell. T cell can stimulate B cell, and that is going to bind to mast cells and basophils. And then when the antigen comes back, it's going to stimulate them to release their histamine and their whatever and cause anaphylaxis or asthma or whatever. Or in the case of TB, type 4 hypersensitivity, the previous one is type 1 hypersensitivity, T cell is going to stimulate macrophages to basically make a granuloma. I don't really know what to say to that. That's with Th1, and that is also producing IFN gamma. You get a lot of IFN gamma production in TB, okay? And all these cells are communicating with cytokines. That's what's stimulating inflammation. That's what's stimulating anti-inflammation. Okay, this was long, but I hope it's kind of helpful.